Funaria is a common type of moss found in damp, shady environments worldwide. With over 210 species, Funaria is characterized by its ability to thrive in various conditions, including disturbed areas and post-fire environments. Unlike vascular plants, Funaria lacks true roots, stems, and leaves. Instead, it has simpler structures that perform similar functions. The name Funaria derives from the Latin word funis, meaning rope, likely referring to its twisted seta or stalk. This moss is frequently used in beginner biology classes due to its relatively simple structure and ease of observation under microscopes. It provides an excellent model for studying plant reproduction and structure. This concludes our introduction to Funaria moss, a fascinating organism that bridges the gap between aquatic algae and vascular land plants. Let's explore the taxonomic classification of Funaria moss. Funaria belongs to the kingdom Plantae, which includes all multicellular plants. It's classified within division Bryophyta, the true mosses. Within Bryophyta, it belongs to class Bryopsida, which contains most mosses. Further classified into subclass Funariidae, it belongs to the order Funarialis, more specifically to the family Funariaceae, and finally to the genus Funaria. This taxonomic hierarchy places Funaria among non-vascular plants that reproduce using spores rather than seeds. Understanding this classification helps contextualize Funaria's evolutionary relationships with other plant groups and its place in the plant kingdom. Now that we understand Funaria's classification, let's examine its global distribution and habitat. Funaria moss has a remarkably wide global distribution. It can be found on every continent except Antarctica, making it truly cosmopolitan in its range. Funaria thrives in a diverse range of environments. It can be found in urban settings, woodlands, and grasslands, showing remarkable adaptability. Funaria moss exhibits extraordinary vertical range in its habitat. It can be found at elevations from just 20 meters all the way up to 2,550 meters above sea level. One of the most distinctive ecological characteristics of Funaria is its preference for recently burnt areas. It thrives in nitrogen-rich soils and is often one of the first species to colonize after fires. Section 4. Gametophyte structure. The dominant phase in Funaria's life cycle is the gametophyte, which is the haploid gamete-producing generation. This structure consists of small, leaf-like structures arranged around a central axis. Let's examine its components closely. Though they resemble leaves, these structures lack vascular tissue. Unlike true leaves in vascular plants, moss leaves have no specialized cells for water and nutrient transport. The gametophyte also features rhizoids, root-like filaments that anchor the plant and absorb water and nutrients. Unlike true roots, rhizoids are simple filaments without complex internal structure. The gametophyte's simple structure demonstrates fundamental plant architecture while highlighting the evolutionary differences from vascular plants. Mosses represent an ancient lineage that diverged before the evolution of specialized vascular tissues. The simple yet effective structure of the Funaria gametophyte demonstrates how mosses have adapted to their environments without the complexity of vascular tissues. The sporophyte generation in Funaria is the diploid, spore-producing phase that grows from the gametophyte. The sporophyte consists of three main parts: the foot, the seta, and the capsule. The foot is embedded in the gametophyte tissue and absorbs nutrients to support the sporophyte's growth. The seta is an elongated stalk that elevates the capsule above the gametophyte for better spore dispersal. The capsule is the pear-shaped structure that produces and contains the spores. The peristome teeth at the top of the capsule are specialized structures that respond to humidity changes, controlling the release of spores. In dry conditions, the teeth curl inward, closing the capsule. In humid conditions, they extend outward, opening the capsule and allowing spore dispersal. The sporophyte's specialized structure efficiently produces and disperses spores, ensuring the continuation of the moss life cycle. Funaria reproduces sexually through specialized structures called antheridia and archegonia. Antheridia are male structures that produce sperm cells. Archegonia are female structures containing a single egg cell. Sexual reproduction in Funaria requires water, highlighting its dependence on moist environments. During fertilization, sperm cells are released from the antheridia and swim through water to reach the egg in the archegonium. After fertilization, the zygote is formed and begins developing into the sporophyte generation. The developing sporophyte remains attached to and nutritionally dependent on the gametophyte. This nutritional dependency is a characteristic feature of bryophytes like Funaria. Funaria moss employs several methods of asexual reproduction to propagate without the fusion of gametes. The first method is fragmentation, where pieces of the moss gametophyte break off and develop into completely new plants. The second method involves specialized structures called gemmae, which form in cup-like receptacles on the moss. These disc-shaped gemmae are dispersed by water or wind and develop into new moss plants. Asexual reproduction provides several evolutionary advantages for Funaria moss. It allows rapid propagation when conditions are favorable and doesn't require water for fertilization like sexual reproduction does. This gives Funaria an advantage in colonizing new areas and persisting through periods of unfavorable conditions. Through these asexual reproduction methods, Funaria moss demonstrates remarkable adaptability to diverse environmental conditions. The life cycle of Funaria moss demonstrates the fundamental alternation of generations patterns seen throughout plant evolution. Let's explore the complete life cycle of Funaria moss, from spore to mature plants and back again. The Funaria life cycle begins with spore germination. The spore develops into a filamentous structure called the protonema. The protonema then develops into the mature gametophyte, which is the dominant phase in the moss life cycle. This leafy green structure is what we typically recognize as moss. The mature gametophyte produces sex organs. Male antheridia produce sperm cells, while female archegonia contain egg cells. Fertilization requires water, as sperm must swim to reach the egg. After fertilization, the zygote develops into the sporophyte while remaining attached to and dependent on the gametophyte. The sporophyte consists of a stalk and a spore-producing capsule. Inside the sporophyte capsule, cells undergo meiosis to produce haploid spores. When conditions are right, the capsule opens and releases these spores. When these spores land in suitable environments, they germinate to form new protonema, which develop into gametophytes, thus completing the alternation of generations cycle. This alternation of generations between haploid gametophyte and diploid sporophyte is a fundamental reproductive pattern observed throughout plant evolution. Funaria moss serves as an early colonizer in disturbed environments, particularly after fires. Despite its small size, Funaria helps stabilize soil and prevent erosion through its rhizoids and growth pattern. Funaria contributes to nutrient cycling in ecosystems by absorbing nutrients from its environment and releasing them when the moss decomposes. Funaria moss provides crucial microhabitats for tiny invertebrates, offering shelter, moisture, and food. Funaria can accumulate heavy metals and other pollutants, making it a valuable bioindicator for environmental pollution. Scientists use Funaria moss for environmental monitoring studies because it provides valuable data on pollution levels in different areas. Despite its small size, Funaria moss plays crucial ecological roles in stabilizing ecosystems and monitoring environmental health.